Hello there, and welcome to Sleepy Science Stories. Tonight we are going somewhere very close. Not across a desert, not through a void in space. Tonight we are going inside. Into the architecture of the cell. Into the molecule that carries the instructions for everything you are. Into a question that science has only recently begun to ask seriously. And that, once asked, changes the way you think about yourself, your parents, your grandparents, and the long chain of people who came before all of them. The question is this: can the things that happened to your ancestors be passed down to you, not as stories, but as biology? The fears they lived through, the hunger they endured, the losses they carried, not as memories you were told, but as patterns in your DNA. Patterns that shape your nervous system, patterns that tune your stress response, patterns that make you more anxious or more resilient or more sensitive to specific kinds of threat because your grandmother was, and her grandmother before her. This is not a metaphor. It is a scientific hypothesis, supported by a growing body of evidence, debated intensely by researchers around the world, and still far from fully resolved. The field is called epigenetics, and what it has found is remarkable enough to deserve a slow and careful examination. In laboratories, in the children of Holocaust survivors, in the offspring of mice trained to fear cherry blossoms. Tonight we will learn what epigenetics is. We will learn how your DNA can be changed, not in its sequence, but in how it is read by the experiences you have in life. We will learn about the experiments that first suggested trauma could be inherited. We will follow the science into human populations and understand what has been found and what remains genuinely uncertain. We will think about what it means for healing, if the wounds of the past live in our cells, and if the cells can change. And we will sit with something that is both unsettling and strangely comforting. The idea that you carry more than your own history, that in the biology of your stress response, your immune system, your emotional tendencies, there may be echoes of lives you never lived, of fears you never faced, of winters you never survived. All of it written not in words, but in the quiet chemistry of the molecule that runs every cell in your body. If you enjoy these sleepy science journeys, please consider liking or subscribing. Every listener helps. For now, let your body settle. Let your breath slow, and let your mind drift inward toward the smallest and most intimate machinery of who you are. We are going into the DNA, and what is in there may surprise you. Take a breath. Feel the weight of your body against whatever is holding you. In this moment, your cells are doing something extraordinary. They are reading your DNA, not all of it, just the parts that are relevant right now, in this tissue, at this hour, with this level of stress hormones in your bloodstream. The rest is silent, wrapped up, covered over with chemical marks that say not now, or not in this cell, or not until the conditions change. Those chemical marks are the epigenome, and they have a history that goes back further than your own birth, further than your parents' birth. They carry, in some small measure, the history of the people who came before all of you. That history is what tonight is about, not as philosophy, as biology, as something you can measure and study and begin to understand. And perhaps, in understanding, begin to change. The science of epigenetic inheritance is one of the most exciting and most debated areas in all of modern biology. It challenges long-held assumptions about what is fixed and what is flexible in our nature. It suggests that the line between experience and biology, between history and genetics, is far less clear than we have been taught. Tonight, we are going to understand why, and what it means for you, for the people who came before you, and for the people who may come after. Before we begin, take a moment to notice your body, the rhythm of your breathing, the weight of your limbs, the slight tension in your shoulders, or the particular quality of calm you may feel right now in the quiet of this moment. All of that, the way your nervous system is calibrated, is shaped by your biology. the threshold at which your heart rate rises, the ease or difficulty with which your body settles into rest. And your biology was shaped not only by your own life. It was shaped, at least in part, by lives that came long before yours. That is the idea at the center of tonight's story. Not as mysticism, not as metaphor, as science, as chemistry, as the specific and measurable behavior of molecules inside the nucleus of every cell in your body. The science is called epigenetics, and what it has been discovering over the past several decades is that the line between biology and biography is thinner than we thought. That experience leaves physical marks on the genome, not changing the letters of the code, but changing how the code is read, and that those marks, written by the experiences of one life, can sometimes be carried forward into the next, and perhaps the next after that. What this means for how we understand ourselves is one of the most profound and most carefully debated questions in modern biology. Our inherited anxieties, our inexplicable sensitivities, our body's responses to the world. Tonight, we explore it together. Slowly, honestly, with full acknowledgement of what is known and what is not. And with the understanding that whatever you find in this story, it does not diminish you. It contextualizes you. It places you in a longer chain than you may have imagined. A chain of biology, of chemistry, of survival. That ends, for now, here, with you. Breathing, present, at rest, at rest. Before we can talk about inherited trauma, we need to understand what DNA is. And, more importantly, what epigenetics changes about it. Most of us learned in school that DNA is the blueprint of life. That your DNA contains the instructions for building every protein, every enzyme, every structural molecule in your body. That it is a long, twisted ladder made of 4 chemical letters, arranged in specific sequences that code for specific proteins, and that this sequence is fixed. You are born with a particular DNA sequence, and you carry it to the end of your life, copied faithfully into every cell. This is mostly true, but it is not the whole story. Your DNA sequence is fixed, but whether any given gene in that sequence is active or silent, whether it is being read by the cellular machinery and producing proteins, that is not fixed. It is regulated. It changes depending on what type of cell you are in, what stage of development you have reached, and what has been happening in your environment. A muscle cell and a nerve cell have identical DNA, but they are completely different kinds of cells, doing completely different things. The reason is that different genes are active in each. The muscle cell is reading certain genes. The nerve cell is reading a different set. The underlying sequence is the same. The readout is different. This process of controlling which genes are read and which are silenced is called gene expression. And the system that controls gene expression without changing the underlying DNA sequence is called epigenetics. The word epigenetics means above genetics, It refers to the layer of chemical information that sits on top of the DNA, telling the cell which parts to use and which to ignore. There are several mechanisms in this system. The most studied is called DNA methylation. DNA methylation works like this. At specific points along the DNA strand, small chemical groups called methyl groups can be attached to the DNA molecule. These methyl groups do not change the sequence of letters. They do not alter the code itself, but they act like sticky notes. They recruit proteins that silence the gene beneath them. A gene with methyl groups attached to its promoter region is turned off. The cell cannot read it. No protein is made. When those methyl groups are removed, the gene can be read again. The gene turns on. Proteins are produced. This on-off switching is happening constantly throughout your body. Different cells use different gene expression patterns. The epigenome, the full collection of epigenetic marks on your DNA, is the instruction manual for which genes to use. Not changing the letters of the book, but determining which chapters are open and which are closed. What makes this relevant to trauma is what happens next. The epigenetic marks, those methyl groups and other chemical modifications, respond to the environment. Not immediately and not simply, but in ways that are measurable and real. Chronic stress changes epigenetic patterns. Starvation changes epigenetic patterns. Early childhood adversity changes epigenetic patterns. The experience of the organism writes itself into the epigenome. And crucially, some of those marks can be inherited. When cells divide, the epigenetic marks on the parent cell can be copied onto the daughter cells. This is how a liver cell stays a liver cell. And in some cases, epigenetic marks survive not just cell division, but reproduction. They pass into the eggs and sperm, and from there, into the next generation. That is the beginning of the story of epigenetic inheritance. That is where the question of ancestral trauma begins. Not in the DNA sequence, in the layer above it, in the sticky notes that tell the genome what to do. The concept of the epigenome is still relatively new. The word epigenetics was coined in the 1940s by the biologist Conrad Waddington. He used it to describe the process by which a single fertilized egg gives rise to many different cell types of a complex organism, all with the same DNA, all doing completely different things. Waddington imagined the process as a landscape, with valleys and ridges, channeling the development of cells into specific stable types. The molecular basis of that landscape, the methyl groups and the histone modifications and the non-coding RNAs, has been worked out over decades of subsequent research. And what emerged from that work was something unexpected. Not just a system for managing cell identity during development, but a system that is continuously responsive to the environment, that reads the conditions the organism is living in, and that adjusts which genes are active accordingly. This responsiveness is the key. It is what makes the epigenome a potential carrier of inherited experience. If experience changes epigenetic marks, and if those marks can survive through reproduction, then the experiences of one generation can, in principle, influence the biology of the next. The mechanism is real. The question is how far it reaches. And tonight, we will follow that question as far as the current science allows. There is also a third mechanism worth mentioning, alongside DNA methylation and histone modification. It is called non-coding RNA. RNA is normally thought of as the messenger molecule, the intermediate between the DNA blueprint and the protein product. But there is a large class of RNA molecules that never get translated into proteins. They regulate other genes instead. They act as switches and dials, turning up or turning down the activity of specific genes without changing the sequence of the DNA. These non-coding RNA molecules are abundant in the reproductive cells, particularly in sperm. They are sensitive to environmental conditions, and research in animals has shown that they can carry information about the parent's stress experience into the fertilized egg, where they influence the development of the offspring. They are, in some sense, the most direct known pathway for epigenetic inheritance. Not methyl marks on the DNA itself, but molecular signals floating in the fluid of the sperm, ready to enter the egg and begin speaking to the offspring's genome, before that genome has even had time to begin reading itself. In 2013, researchers at Emory University published a study that made headlines around the world. The experiment was simple in design, striking in its results. The researchers took male mice. They trained them to fear a specific smell. The smell was acetophenone, a sweet, cherry blossom-like fragrance. The training was done by pairing the smell with a mild electric shock to the feet. After repeated pairings, the mice learned to fear the smell. Every time they encountered it, they froze. Their stress response activated. Their bodies prepared for danger. This is standard classical conditioning. Pavlov with mice and smell instead of dogs and bells. Nothing unusual so far. What happened next is what changed everything. The trained mice were allowed to breed with naive females who had never encountered the smell or the shock. The resulting offspring were raised without any contact with the father. No chance for learned behavior to be transmitted directly. The baby mice were born, raised, and then tested with the cherry blossom smell. They were afraid of it. Not dramatically. Not paralyzed. But measurably, reproducibly more sensitive to that specific smell than control mice, whose fathers had not been conditioned. Their threshold for detecting the smell was lower. Their physiological response to it was stronger. Their olfactory neurons, the nerve cells that detect smell, were more numerous and more developed in the specific area dedicated to detecting acetophenone. The researchers then went further. They looked at the sperm of the trained mice. They found epigenetic changes in the gene responsible for detecting acetophenone. Specifically, they found reduced methylation, which means that gene was more active in the sperm than it was in the sperm of untrained mice. The gene had been turned up by the fear experience. That elevated gene activity, encoded in the sperm, appears to have been passed to the offspring. The offspring inherited not just the DNA, but the epigenetic mark that the father's fear had written onto it. And as a result, the offspring were born with a nervous system already primed to be sensitive to a smell their father had learned to fear. The grandsons of the trained mice showed the same effect. 3 generations connected by a chemical mark on a single gene, written by a single fearful experience in the grandfather's life. The implications were extraordinary. If a mouse can inherit its grandfather's fear of a smell, what else can be inherited? And does the same mechanism operate in humans? Those questions sent researchers into archives, into population studies, into the clinical records of trauma survivors and their children. And what they found was more complex than the mouse study, and more cautious in its conclusions, but also more human, and in some ways more haunting. Let that settle. A grandfather mouse, trained to fear a cherry blossom. His son, who never smelled it in fear, afraid of it anyway. His grandson, 2 generations removed from the original shock, still carrying in his olfactory system the trace of something that happened before he was born. A ghost in the biology, a memory that was never his, written in the chemistry of his nerves and his sperm. That is where we begin the human story. The cherry blossom study was published in a journal called Nature Neuroscience. When it appeared, it generated enormous attention, both within the scientific community and in the general press. It was striking, reproducible, and mechanistically plausible. It was also limited, in the way animal studies always are, in what it could tell us about humans. Mice are not humans. The olfactory system of a mouse is far more prominent and more directly connected to fear circuits than the olfactory system of a human being. The training involved a very specific and measurable sensory stimulus, paired with a very clear and controllable stressor. Human trauma is none of those things. It is complex, varied, and deeply entangled with social context, language, and meaning. But the mouse study proved a principle. It showed that a specific learned fear could write an epigenetic mark in the reproductive cells, and that mark could influence the nervous system of an offspring who had never had the original experience. The principle is there. The question is whether it operates in humans, at the scale of real historical trauma, in the messy and uncontrollable conditions of actual human lives. That question sent researchers to some of the most documented examples of mass human trauma in history. And among those, the most studied, is one of the most devastating: the systematic murder of 6 million Jewish people in Europe during the Second World War, and what happened to the biology of the people who survived, and their children, in the decades that followed. But before we go there, it is worth pausing on what the cherry blossom study actually proved, and what it did not. It proved that a specific, measurable fear could be transmitted from father to offspring through a biological mechanism. The researchers ruled out behavioral transmission by separating the father from the offspring entirely. They identified epigenetic changes in the sperm that corresponded to the behavioral changes in the offspring. They replicated the findings. The study was methodologically rigorous. What it did not prove is that all fears, or all traumas, or all significant experiences are transmitted this way. The cherry blossom study worked because olfactory fear conditioning is a very specific and very measurable phenomenon in mice. The olfactory system is ancient, well-mapped, and directly connected to fear circuits in the brain. Transmitting a sensitivity to a specific smell through the germline is a much simpler biological task than transmitting the complex, multidimensional experience of human trauma. Nevertheless, the study opened a door. It showed that the germline is not sealed against the experiences of the organism, that something from the life of the parent can reach the reproductive cells, and from there, reach the next generation. That principle is now supported by multiple independent lines of evidence in multiple species, using multiple different kinds of stress and multiple different measures of transmission. The year is 1944. The Netherlands is under German occupation. In September, the Dutch railway workers went on strike to support the Allied liberation effort. In retaliation, the German authorities cut off food and fuel supplies to the Western Netherlands. What followed was one of the worst famines in modern European history. It is called the Dutch Hunger Winter. From late 1944 into the spring of 1945, roughly 4.5 million people were trapped in a region. Daily food rations fell to as low as 400 calories. The normal requirement for an adult is around 2,000. People ate tulip bulbs. They burned furniture for heat. Tens of thousands died. The famine ended when the Allies liberated the Netherlands in May 1945, but its effects did not end with liberation. Decades later, researchers began studying the children of women who had been pregnant during the hunger winter. They found something remarkable. The children who had been in the womb during the famine, particularly during the first trimester, showed elevated rates of heart disease, obesity, type 2 diabetes, and schizophrenia. Compared to siblings born before or after the famine, they were measurably different in their metabolic and health profiles. This was already significant. But then researchers looked deeper. They looked at the epigenomes of these children, now adults in their 50s and 60s. They found differences in DNA methylation at specific genes compared to their siblings who had not been exposed to famine in the womb. Genes related to insulin regulation, to growth, to metabolism, all showing altered patterns of methylation, consistent with the health differences the researchers observed. The experience of the famine had written itself into the epigenomes of the developing fetuses, and those epigenetic marks had persisted for more than 60 years. They were still there, readable in the blood and tissue of people who were now grandparents themselves. Then researchers looked at the grandchildren, the children of the famine children, and they too showed measurable differences. Smaller, more subtle, but detectable. The biological echo of a famine that ended before they were born, before their parents were grown, reaching forward through 2 generations of biology. The Dutch Hunger Winter was not a study of emotional trauma. It was a study of nutritional and physical stress, but it established a foundational principle. The environment a parent or grandparent survives can alter the biology of descendants who never experienced it. The mechanism is epigenetic. The scope is multigenerational. The evidence is some of the most compelling in the field. And it raised the next question. If famine leaves this kind of biological inheritance, what about trauma? What about the experience of surviving something terrible? That is psychological rather than purely physical? What about the Holocaust? The Dutch Hunger Winter is particularly valuable as a scientific case study because of its unusual properties. It affected a large, relatively well-documented population. It had a clear start and end date. And because the Netherlands has excellent historical health records, researchers could track the offspring of famine survivors across decades with unusual precision. The findings were not subtle. Children who had been in the womb during the famine showed differences that persisted into old age. Their epigenomes carried marks that were still detectable 60 years later, and those marks corresponded to real differences in health outcomes. Not random variation, not noise. A consistent biological signal linking the experience of starvation in the womb to the function of metabolic genes decades later. This is sometimes called prenatal programming. The idea that the environment a fetus develops in shapes the way its genes will function throughout life. The Dutch hunger winter offered one of the clearest natural experiments ever available to test this idea in humans, and the results have been replicated and extended in subsequent studies of other famines, including the Chinese famine of 1959 to 1961, and famines in parts of Africa. Each of these studies adds evidence to the same general picture. What a pregnant woman experiences writes biological marks into the developing fetus. Those marks can persist for life, and they may be transmitted in some diminished form to the next generation after that. Not because the DNA changed, but because the epigenome, the layer of chemical instruction above the DNA, was set differently by the conditions of development. The Dutch hunger winter findings were important for another reason too. They demonstrated that the timing of exposure matters enormously. Children who were in the womb during the first trimester of the famine showed different effects than children who were in the womb during the third trimester. The early stages of fetal development appear to be particularly sensitive periods for epigenetic programming. Disruptions during those windows leave different marks than disruptions later in development. This sensitivity to timing is consistent with what researchers have found in other contexts. Early childhood, particularly the first 2 years of life, is another window of heightened epigenetic sensitivity. Experiences of stress, deprivation, or inconsistent care during those years produce measurable epigenetic changes that persist into adulthood. The developing organism is not passive. It is actively reading its environment and using that information to set the biological defaults it will carry through life. This is both sobering and important to understand. It is sobering because it means that adverse experiences during sensitive developmental windows can have very long-lasting biological consequences. But it is important because it tells us where interventions might be most powerful. Protecting pregnant mothers from severe stress. Providing consistent, warm, responsive care in early childhood. Ensuring that the environments where children develop are safe and adequately nourishing. These are not just good ideas. They are biologically consequential. They shape epigenomes. They influence the long-term health and stress biology of the people those children will become. And possibly, to some degree, the people their children will become after them. The biology of the next generation begins, in some measure, in the conditions we create for this one. Rachel Yehuda is a neuroscientist and trauma researcher at the Icahn School of Medicine at Mount Sinai in New York. For decades, she has been studying the biology of trauma, particularly in Holocaust survivors and their children. What she found, over years of careful measurement, was unexpected. The children of Holocaust survivors showed abnormal levels of cortisol. Cortisol is the primary stress hormone. It is released by the adrenal glands in response to threat. It prepares the body for fight or flight. It elevates heart rate, mobilizes energy, sharpens attention. In normal function, it rises in response to stress and then falls back to baseline when the threat passes. In Holocaust survivors themselves, Yehuda had previously found that cortisol levels were chronically low. This is counterintuitive. You might expect trauma survivors to have elevated cortisol, but in chronic PTSD, the system becomes dysregulated in the opposite direction. The feedback loop becomes hypersensitive. The body reacts intensely to small stressors, even as baseline levels remain low. This pattern, low baseline cortisol with heightened sensitivity, is a known marker of PTSD. Yehuda found the same pattern in the children of Holocaust survivors. People who had not themselves experienced the Holocaust. people who had grown up in safety. But people whose cortisol regulation was measurably different from the children of parents who had not experienced the Holocaust. In a study published in 2015, Yehuda's team measured methylation at a specific site on a gene called FKBP5. This gene is involved in regulating the glucocorticoid receptor, which controls how the body responds to cortisol. The methylation pattern at this site was different in Holocaust survivors. And it was different in their children, in the same direction. The children showed epigenetic changes in the same gene, in the same location, correlated with parental PTSD. The sex of the traumatized parent mattered. Children with mothers who had PTSD showed different patterns than children with fathers who had PTSD. In some cases, the effects went in opposite directions depending on which parent carried the PTSD diagnosis. The biology was not simple, but it was measurable. Yehuda herself has been careful about the conclusions. She has publicly said it would be premature to claim that epigenetic inheritance definitively explains these findings. The study involved relatively small numbers, controlling for environmental transmission, for parenting style, for social learning, is extraordinarily difficult in human studies. You cannot take a child away from their parents after birth and raise them in a controlled environment the way researchers do with mice. But the patterns are there. The epigenetic differences are there. And they are consistent with what the mouse studies and the famine studies predict. Something is being transmitted. The debate is about what exactly and through exactly which mechanisms. What is certain is that the children of Holocaust survivors are biologically different from the children of parents who did not experience the Holocaust in specific and measurable ways. Their cortisol systems are different. Their gene methylation patterns are different. Their stress responses are different. These are not imagined differences. They are measured in blood and tissue. What is uncertain is the pathway. Was the epigenetic change in the FKBP5 gene truly transmitted through the germline, through the sperm or egg? Or did the parent's PTSD alter the intrauterine environment during pregnancy, producing epigenetic changes in the developing fetus through prenatal programming? Or did the parent's altered parenting behavior, shaped by trauma, create early life stress in the child, which then altered the child's epigenome through the child's own stress response? All three pathways could produce the same biological outcome in the offspring. All three involve epigenetics. But only the first is truly epigenetic inheritance in the strict biological sense. Untangling these three pathways in humans is the central scientific challenge. And it is why researchers like Yehuda, despite their extraordinary and carefully collected data, are careful to say that the evidence is suggestive rather than conclusive. The question is alive. The research is ongoing, and the findings, careful and partial as they are already changing the way medicine and psychology think about the long shadow of trauma. They are changing clinical practice. Mental health practitioners working with the children and grandchildren of trauma survivors are beginning to understand something important. The biological sensitivity they observe in their clients may have roots that go beyond the client's own history. The heightened startle response, the difficulty regulating emotion, the pervasive sense of danger in seemingly safe situations, that the nervous system they inherited was calibrated, in part, by experiences that happened before they were born. This changes the therapeutic relationship in subtle but important ways. Instead of asking only what happened to you, the clinician might also ask what happened to the people who raised you and to the people who raised them, not as an exercise in blame, but as a way of understanding the biological context from which the client's responses emerged. The anxiety is not a character flaw. It is a biological inheritance, an inheritance that can be understood, worked with, and over time, modified. The Holocaust survivor research has also raised important questions about how trauma affects different sexes differently. Yehuda's work found that the effects of maternal PTSD and paternal PTSD on children were not the same. The sex of the traumatized parent and the sex of the child both appeared to influence the direction and magnitude of the epigenetic changes observed. This complexity is not surprising, given that the stress system operates differently in males and females. But it adds another layer of nuance to the already complex picture. The inheritance is not uniform. It is shaped by who carried the trauma and who received it. This complexity does not weaken the finding. It deepens it. It suggests that the biology of trauma transmission is not a simple, single mechanism. It is a web of interacting effects, shaped by sex, by developmental timing, by the specific nature of the trauma, and by the relationship between parent and child. Understanding that web, in its full complexity, is one of the defining scientific challenges of the coming decades. One of the most debated questions in this field is how epigenetic information gets from the parent's experience into the reproductive cells. This transfer is not obvious. It requires a pathway from the body's somatic cells to the germline, from the neurons that process stress, or the endocrine glands that release stress hormones, to the eggs or sperm that will one day become the next person. Your body has two types of cells: somatic cells, which make up your organs and tissues, and germline cells, which become your eggs or sperm. Epigenetic changes in your somatic cells stay in your somatic cells. If stress changes the methylation of genes in your brain, that change is not automatically transmitted to your sperm. For epigenetic inheritance to work, there has to be a mechanism by which what happens in the body somehow reaches and alters the germline. That pathway is not fully understood, but research has identified several candidates. One pathway involves small non-coding RNA molecules. RNA is the messenger molecule that carries instructions from DNA to the protein-making machinery. But there is a class of RNA molecules that do not code for proteins. They regulate gene expression instead. These molecules are abundant in sperm, and they are sensitive to environmental conditions. Studies in mice have shown that stress changes the profile of small RNAs in sperm. When a mouse experiences chronic unpredictable stress, its sperm contain a different mix of small RNA molecules than the sperm of unstressed mice. When those altered sperm are used to fertilize eggs, the offspring show behavioral differences. More anxiety-like behavior, different stress hormone patterns. The small RNAs in the sperm appear to be carrying information about the father's stress experience into the developing embryo. In one experiment, researchers extracted the small RNAs from stressed mice's sperm and injected them into fertilized eggs from completely unstressed parents. The offspring of those eggs showed the same stress-related behavioral changes as the offspring of the stressed mice. The RNA alone was sufficient to transmit the effect, without any DNA change, without any behavioral interaction, just the RNA from the sperm of a stressed animal carrying a message about that animal's experience into the next generation. This is among the most compelling mechanistic evidence for epigenetic inheritance of stress effects. It bypasses the objection that the effects might be due to altered parenting. The offspring never had contact with the stressed parent. The only connection was the small RNA molecules in the sperm. Maternal transmission is even more direct in some ways. A mother carries her eggs from birth, and a developing fetus is exposed to everything in the mother's bloodstream, filtered through the placenta. Maternal stress hormones cross the placenta. They alter fetal development. They change the epigenetics of the developing brain. This is sometimes called prenatal programming, and it is well established. The child of a mother who experienced severe stress during pregnancy shows measurable biological differences from a child whose mother did not. The debate is not whether these effects exist. They clearly do. The debate is about which effects are truly transmitted through the germline versus which are the result of the uterine environment during pregnancy, versus which are the result of parenting behavior and social transmission after birth. Untangling those three pathways in humans, without the controlled experimental conditions available in animal studies, is the central methodological challenge of this field, and it is why the science remains genuinely uncertain, even as the evidence accumulates. But the animal evidence for germline transmission is strong enough that researchers continue to investigate the possibility seriously in humans. Studies of Vietnam veterans with PTSD have found differences in DNA methylation in their sperm at regions associated with psychiatric vulnerability. Those methylation patterns correlate with mental health diagnoses in the veterans' children. The correlation is not proof of causation, but it is consistent with the hypothesis. Similar work has been done with children whose mothers experienced severe stress during pregnancy, and with populations where stress during the preconception period can be identified and measured. In each case, the offspring show biological differences, and in each case, the question of which pathway produced those differences remains only partially answered. What the research does establish clearly is that the stress of one generation reaches into the biology of the next through multiple channels simultaneously. The germline, the womb, and the parenting environment all appear to play roles, and all three involve epigenetic changes in the offspring. Understanding how much each pathway contributes in different circumstances is the work of the coming decades of research. What is already clear from the existing evidence is that stress during the preconception period, meaning before pregnancy begins, can affect the offspring through the germline. This is particularly well documented in paternal transmission. A father who experiences severe stress in the months before conception shows changes in the small RNA content of his sperm. Those changes correspond to behavioral and biological differences in his offspring, and since the father's only biological contribution to the offspring is the sperm, the prenatal environment and early parenting behavior cannot explain these effects. The transmission has to be running through the sperm itself. Isabel Mansai of the University of Zurich has spent years documenting this in mice. She has shown that early life trauma in male mice, produced by unpredictable maternal separation, produces behavioral and metabolic changes that can be detected across multiple generations. She has isolated the non-coding RNA in the sperm of traumatized males and injected those RNAs into fertilized eggs from normal, untraumatized mice. The offspring showed the same behavioral changes as offspring of traumatized fathers. The RNA alone is sufficient to transmit the effect. No DNA change, no behavioral interaction, just a collection of small molecules from the sperm of a stressed animal carrying the signal of that stress into the next generation. This is among the most mechanistically compelling evidence in the field, and it has driven a wave of interest in understanding which specific RNA molecules are involved, what they do to the developing embryo, and how their content is determined by the parent's experience. The evidence for epigenetic inheritance of trauma is striking, but science requires skepticism, and the skeptics of this field raise important points that deserve honest engagement. The first and most fundamental challenge is the issue of epigenetic reprogramming. When a sperm fertilizes an egg, the embryo undergoes a massive wave of epigenetic reprogramming. The epigenetic marks from both parents are largely erased, and a new epigenetic landscape is established, specific to the developing embryo. This erasure is thought to serve an important biological function. It allows the embryo to start fresh, to develop its own cell-type specific epigenome, without being locked into the epigenetic state of the parent cells. If most epigenetic marks are erased during this reprogramming, how can trauma-associated marks survive to influence the offspring? The answer is that the reprogramming is not complete. Some marks escape erasure, particularly at regions of the genome called imprinted genes, which have marks that deliberately resist reprogramming. But whether trauma-associated marks specifically escape erasure in a consistent and heritable way is not yet established in humans. The second challenge is the problem of social transmission. Humans are social animals. We transmit enormous amounts of information to our children through behavior. A parent with PTSD parents differently than a parent without PTSD. They are more anxious, more vigilant, more reactive to perceived threats. This affects the child's development, It changes the child's stress response. It can alter the child's epigenome through the child's own experiences of stress and inconsistent parenting. This is not epigenetic inheritance. It is social learning, but it produces similar biological outcomes. And in human studies, separating these pathways is extraordinarily difficult. You cannot run the equivalent of the mouse cross-fostering experiments, where pups are raised by mothers who did not experience the stress. The third challenge is sample size and replication. Many of the most cited human studies in this field involve small numbers of participants. The Holocaust survivor study by Yehuda involved 32 survivors. Results from small samples need to be replicated in larger, independent studies. Some findings in this area have proven difficult to replicate. The history of science is full of small sample results that look striking and turned out to be artifacts of noise. Yehuda herself, one of the most prominent researchers in this field, has acknowledged these limitations publicly. She has said that the media coverage of early findings was overblown, that the popular narrative of epigenetic trauma inheritance as a certain and well-established fact outran the actual state of the evidence. The honest scientific position is this: something is being transmitted across generations in the children of trauma survivors. The biological signatures are real. The mechanisms are partially understood. But whether those mechanisms involve germline epigenetic inheritance rather than prenatal environment or social transmission has not been conclusively established in humans. The science is real. The caution is also real. Both deserve respect. And there is one more reason for caution, worth naming directly. The popular narrative of epigenetic trauma inheritance can become its own kind of burden. If people believe their biology has been permanently damaged by their ancestors' suffering, they may feel hopeless. And that hopelessness is not warranted by the actual science. The evidence suggests that epigenetic marks respond to experience throughout life, that they can change, that the inheritance is not a sentence. And a responsible account of this science needs to hold both things at once. The marks are real, and the marks can shift. The past is in you, and the present is also in you. Both are true. Both matter. There is a fourth reason for scientific caution that is worth naming clearly. The biological complexity of epigenetic inheritance in humans is genuinely staggering. The human genome contains roughly 20,000 protein-coding genes. Each gene has multiple potential methylation sites. The interactions between different epigenetic marks, and between those marks and the underlying DNA sequence, and between the epigenome and the broader cellular environment, are almost incomprehensibly complex. Animal studies can control for much of this complexity by using inbred strains of mice with nearly identical genomes. Human populations are genetically diverse. That diversity means that the same epigenetic change may have very different effects in different people, depending on the genetic background it is operating in. A methylation change at a stress-regulating gene that is harmful in one person's genome might be neutral or even beneficial in another's. This genetic diversity makes it very difficult to identify clean epigenetic signals in human studies. The noise is enormous. The sample sizes required to detect real effects amid that noise are very large. And many of the early human studies in this field were not large enough to be confident that their findings would hold up across different populations. This does not mean the findings are wrong. It means they need to be replicated in larger samples across diverse populations before they can be treated as established facts. That work is underway. The field is maturing. The methods are improving. Larger studies are being conducted, and the results, as they accumulate, will either strengthen or revise the picture that has emerged from the earlier, smaller work. Science is a process, not a set of conclusions. The story of epigenetic trauma inheritance is still being written, and the people writing it are doing so with appropriate care and appropriate humility. The popular version of the story, which often presents inherited trauma as a confirmed and well-understood fact, is ahead of the actual evidence. But the evidence itself is real and growing. and pointing in a consistent direction. The direction of connection. Across time, across generations, through the chemistry of the cell. And in that direction, there is something genuinely new, not just in science, but in how human beings can understand their own nature. For most of history, the self has been understood as beginning with birth, or perhaps with conception. What epigenetic inheritance suggests is that the self begins earlier, that you arrive already in conversation with the past, already shaped by it, already carrying something of it, not as burden alone, but as continuity, as belonging, as the latest chapter in a story that did not begin with you and will not end with you either. Most popular accounts of epigenetic trauma inheritance frame the story as one of damage. The trauma of the ancestor injures the descendant. The genes are haunted. The biology is scarred. But there is another way to interpret the same findings. A way that is both scientifically interesting and psychologically different in its implications. Consider what the inherited biological changes actually do. In the children of Holocaust survivors, the stress system is more sensitive. Cortisol regulation is altered. The glucocorticoid receptor functions differently. These are often described as vulnerabilities, as increased risk for anxiety and PTSD. But what if you reframe the question? Instead of asking what went wrong, ask what the biology is preparing the offspring for. The parent survived something terrible. The parent's body learned from that survival. The epigenetic changes that resulted are, in some sense, a record of what the environment demanded. A more sensitive stress response. A faster activation of fight or flight. A body that is tuned, at a biological level, to be alert to danger. In a safe environment, that tuning looks like vulnerability. Higher anxiety, stronger response to small stressors. Difficulty calming down after threat. These are real costs. But if the offspring were born into the same dangerous environment as the parent, that tuning would be an advantage. A more sensitive stress system would allow faster detection of threat. A body already primed for danger would respond more effectively. The inheritance, reframed, is not damage, but preparation. A biological message from parent to child. The world is dangerous. Your nervous system should be ready. Yehuda herself has explored this reframing. She has written that it would be a mistake to interpret epigenetic inheritance as always being a story of harm. It may, in many cases, represent a form of biological adaptation, a mechanism by which survival lessons are passed from parent to child, without requiring the child to learn them from direct experience. The same logic applies to the Dutch hunger winter findings. The children of famine survivors showed altered metabolism, more efficient storage of energy, greater propensity for weight gain. In a world where food is plentiful, this looks like a risk factor for obesity and diabetes. But in the world their parents survived, the same biology would have been life-saving. The epigenetic inheritance is, in this view, a kind of biological forecasting. The parent's body making predictions about what the offspring will need, based on what the parent experienced. When the prediction is wrong, when the offspring are born into safety rather than danger, the inheritance can become a burden. But the mechanism itself is not broken. It is doing exactly what it evolved to do. This does not minimize the suffering of people who carry the biological legacy of ancestral trauma. The anxiety is real. The altered stress response is real. The elevated risk is real. But the reframing matters for how we understand it, and it matters for healing. There is also a third layer to this question, beyond damage and adaptation. The epigenetic inheritance of a parent's experience may, in some cases, simply be information, not harmful information, not beneficial information, just a biological signal about the environment, a way for the body to ask, before the offspring is even born, what kind of world am I entering? Is food scarce? Is danger frequent? Is calm a reasonable expectation, or should I be on alert? The offspring's developing biology uses whatever signals are available to set its defaults: cortisol sensitivity, metabolic efficiency, stress reactivity. These are not permanent settings, but they are starting points, and the starting points come partly from the parent's lived experience, transmitted through the chemistry of their reproductive cells. This is not so different from other forms of biological preparation. A mother bird calls to her eggs in a way that prepares the chicks for the acoustic environment they will hatch into. A tree's seeds carry chemical information about the light conditions the parent grew in. Organisms have always prepared their offspring for the world as they know it. Epigenetic inheritance may be one more version of this ancient biological conversation. Older than language, older than behavior, written in the chemistry of the cell itself. And there is something worth sitting with in that image. The idea that preparation for the world begins before birth, that the conditions a parent faced, the dangers they navigated, the shortages they survived, are registered by the body and passed forward, in some form, to the children who will face the next version of those conditions. This is consistent with the broader logic of evolutionary biology. Organisms that can prepare their offspring for the specific environment they are likely to encounter have an advantage over organisms that cannot. If the mechanism is fast enough to operate within a generation or two, it allows populations to adapt to changing conditions more rapidly than natural selection alone would permit. Natural selection works across many generations. Epigenetic inheritance can work across one or two. This possibility is genuinely exciting to evolutionary biologists. It represents a partial rehabilitation of an idea long discredited in biology. Lamarck, the 19th century French naturalist, proposed that organisms could pass acquired characteristics to their offspring. A giraffe that stretched its neck would have offspring with slightly longer necks. Darwin's mechanism of natural selection, operating on random genetic variation, eventually replaced Lamarck's framework. For over a century, the inheritance of acquired characteristics was considered a biological impossibility. Epigenetics has reopened that question, not in the crude form Lamarck imagined. Giraffes do not literally pass stretched necks to their offspring, but the general principle that what happens to an organism during its lifetime can influence the biology of its descendants is now supported by real experimental evidence. In this sense, epigenetic inheritance is a partial vindication of something that was dismissed as unscientific for more than 100 years. Not Lamarck's exact mechanism, but Lamarck's intuition that biology and biography are not as separate as the modern synthesis once insisted. That experience shapes inheritance. That what a life demands leaves a mark that outlasts the life itself. If trauma can write itself into the epigenome, one of the most important questions follows. Can those marks be erased or rewritten? Can the inherited biology be changed? The answer, based on current evidence, is yes. At least partially. At least in some circumstances, epigenetic marks are not as permanent as DNA sequence. They can be added and removed throughout a lifetime. The same methyl groups that silence genes can be removed by other enzymes. The epigenome is, in principle, reversible. That is one of its defining properties. This distinguishes epigenetic inheritance from genetic mutation, which is far more difficult to reverse. Research in animals has already demonstrated that the epigenetic effects of early stress can be modified by later experience. Isabel Mansi, a neuroscientist at the University of Zurich, studied mice that had been traumatized early in life. These mice showed the expected stress-related behavioral changes, and their offspring inherited altered epigenetic marks. But when the traumatized mice were placed in enriched environments with running wheels, toys, social contact, and novel objects, their behavior improved. And critically, their offspring also showed fewer stress-related symptoms. The enriched environment appeared to partially reverse the epigenetic legacy of early trauma. In humans, Several interventions are known to alter epigenetic patterns. Physical exercise changes methylation at genes related to metabolism and inflammation. Psychotherapy changes epigenetic marks associated with stress response. Some pharmaceutical drugs directly target the enzymes that add or remove methyl groups. These drugs are already used clinically for certain cancers, where abnormal methylation contributes to disease. This opens a genuinely hopeful dimension to the story. If trauma writes its marks epigenetically, and if those marks can be modified by experience and intervention, then the inherited biology of trauma is not destiny. It is a starting point, a biological tendency shaped by what came before, but not fixed beyond change. The body is always reading its environment and adjusting. The epigenome is always responding. The marks that arrived with you, inherited from the people who came before, can be written over by the experiences you have now. Not instantly, not completely, not without effort, but they can change. There is something deeply important in that. The past is in you, but the past does not determine you. The chemistry of your stress response was shaped by your ancestors, but it is also being shaped right now by everything you do. Every relationship you have, every safe night of sleep, every experience of being cared for, challenged, or delighted. The epigenome is not a record of the past alone. It is a living, responsive archive, and archives can be updated. The reversibility of epigenetic marks is one of the most therapeutically exciting aspects of this science, because if marks can be added by negative experience, they can also be modified by positive experience. The nervous system that was tuned toward anxiety by inherited stress patterns can be retuned. Not instantly, not without effort, but measurably. This is consistent with what practitioners of trauma therapy have observed for decades. That effective therapy changes something fundamental in the person, not just their thoughts about the past, but the way their body responds to the present. The startle response decreases. The baseline heart rate lowers. Sleep improves. The body, which was set to a level of vigilance appropriate for a more dangerous world, gradually learns that the current world is safer. Epigenetics suggests that some of this change may be happening at the level of gene expression, that the safe environment the therapist helps create, the relationship that provides consistent calm, the physical rest that reduces cortisol, may be writing new marks alongside the old ones, not erasing the inheritance, but adding to it, giving the epigenome more recent and more relevant information about the conditions of the present life. This is why the science of epigenetic inheritance, for all its complexity and uncertainty, is ultimately a hopeful story. The biology is responsive, the past is carried but not fixed, and every experience of safety, connection, and genuine rest is contributing something real at the level of the cell to the ongoing project of becoming. The responsiveness of the epigenome is also why the field of epigenetic medicine is developing so rapidly. Researchers are working to identify specific epigenetic marks that are associated with specific health conditions: depression, anxiety, cardiovascular disease, certain cancers. The goal is not just to identify the marks, but to find ways to reverse them, to use drugs or behavioral interventions, or combinations of both, to write new marks over the old ones. Some of this work is already in clinical use. Drugs called DNMT inhibitors, which reduce DNA methylation, are approved for treating certain cancers where genes have been inappropriately silenced. HDAC inhibitors, which alter histone modification, are similarly in clinical use. These drugs are blunt instruments, affecting methylation or histone modification broadly across the genome. But they are proof of principle. The epigenome can be pharmacologically modified. The marks can be changed from outside the cell. Future interventions will likely be far more targeted. Researchers are working on tools that can add or remove methylation at specific genes, leaving the rest of the epigenome untouched. The precision required is extraordinary, but the direction of travel is clear. Epigenetic medicine is moving toward the ability to specifically address the marks that underlie specific conditions, including, perhaps, the marks associated with inherited stress and trauma. That is a long way in the future. But the direction is set, and the foundation for it, laid by the basic research we have been exploring tonight, is real. There is also a simpler and more immediate version of this hope. You do not need to wait for epigenetic medicine to begin changing your epigenome. It is changing right now. Every night of deep sleep is writing marks. Every morning walk. Every meal eaten without stress. Every conversation that ends in genuine understanding. Every moment you allow yourself to feel safe and to believe it. The epigenome is always listening. It is always updating its picture of the world you live in, and every genuine experience of safety and warmth and rest is a data point it uses to revise what it inherited from the past. The science of epigenetic inheritance is still being written. The debate is ongoing. The evidence is real but incomplete. But even in its incomplete state, this science changes something in the way a thoughtful person can understand themselves. Consider what it means to know that your biology has been shaped by lives you never lived. Most of us carry things we cannot fully explain. Anxieties that seem disproportionate to our current circumstances. Fears that appear without obvious cause. Responses to stress that feel ancient, familiar, and not entirely ours. Patterns of relating to the world that seem to come from somewhere before our own experience began. Psychology has long recognized this. Family systems therapy, developed by Murray Bowen and others, has described for decades how patterns of coping and relating pass through generations, how the trauma of one generation becomes the anxiety of the next, and the depression of the generation after that. Not through deliberate transmission, but through behavior, through parenting, through the subtle shaping of a child's nervous system by the nervous system of the parent. Epigenetics adds a biological layer to this understanding. It says that the transmission is not only behavioral, it is also chemical, that the marks are carried not only in stories and habits and family patterns, but in the DNA itself, in the methylation of genes that regulate how you respond to threat, in the small RNA molecules that were in the sperm or egg from which you came. This changes the relationship between the past and the present. It makes the past more present, more physically intimate, more real. Your grandmother's fear is not just a story you were told. It may be a pattern in the activation of your glucocorticoid receptor. Your great-grandfather's hunger is not just history. It may be a tendency in your metabolism. The people who came before you are not entirely gone. They are, in a biological sense, still here, in you, in the way your body responds to the world. This is a profound shift in how we understand identity. We tend to think of ourselves as originating with ourselves, as beginning with our own birth and our own experiences. The science of epigenetic inheritance suggests otherwise. We begin earlier. We carry a biological inheritance that is not just our genes, but the environmental history of our genes, the experiences of those who came before us, written into the chemistry of the molecule that runs every cell in our body. That inheritance is real. It is partial and probabilistic, not deterministic. It does not make you a prisoner of your ancestors' suffering, but it connects you to them in a way that is more concrete than story, more physical than memory. You carry them in your biology, in your stress response, in your nervous system, in the way your cells read the world. And knowing that, it becomes possible to hold the past with more compassion, both for yourself and for the people whose choices and suffering shaped what you carry. The person who seems inexplicably anxious in situations that seem safe to others. The person whose stress response activates at a lower threshold than seems warranted. The person who struggles to feel calm, even in the presence of genuine safety. These responses may not be personal failures. They may be biological inheritances, patterns set before birth, written in the early experiences that shape the developing nervous system. Based on the lives of people who are no longer here to explain why they passed what they passed, understanding this does not excuse those patterns. It does not make them appropriate to every situation, but it creates a different relationship to them. Instead of experiencing the anxiety as a character flaw, it becomes possible to see it as a message from the past. A message that was sincere when it was sent. A message that may no longer apply, but a message that can be acknowledged and perhaps, over time, gently updated. That is a very different relationship to the self than the one most people have been taught. It is a relationship that includes not just your own history, but the history that arrived with you. And that, in its own quiet way, is a kind of belonging, a connection to the people who came before, written not in memory but in biology, not in language but in the chemistry of the cell. That connection is worth holding with both honesty and gentleness. Honesty, because the science is incomplete, because the mechanisms are not fully understood, because what is certain and what is speculative have not always been clearly distinguished in the popular accounts of this field. The inheritance is real in some measure. How much, and through exactly which pathways, remains genuinely open. And gentleness, because the implications for how we understand ourselves are significant. To know that your nervous system was calibrated, in part, by experiences that preceded your birth, is to change the relationship between yourself and your own biology. It becomes less personal, less like a verdict on your character, more like context, more like something to be understood, worked with, and gradually updated, rather than overcome or denied. There is also a broader social dimension worth noting. Epigenetic research on inherited trauma has been particularly important for communities whose historical suffering has sometimes been dismissed or minimized. Indigenous communities, whose experiences of colonial violence and displacement have biological echoes in health disparities that persist for generations. African American communities, whose experiences of enslavement and ongoing structural racism produce biological effects visible in stress hormone patterns and health outcomes. Jewish communities, whose Holocaust history is now being studied in the most rigorous biological terms available. The science says something important to all of these communities: that the suffering was real, that it left real marks, that those marks persist in biological form in the people who came after, and that healing those marks truly and completely requires acknowledging the suffering that created them. We've come a long way tonight, from the outside of the cell to the inside of the nucleus, from the concept of DNA as a fixed blueprint to the understanding of the epigenome as a living, responsive layer of information, shaped by experience, transmissible across generations, revisable by new experience. We followed the cherry blossom mice, a grandfather's learned fear, encoded in the sperm, carried to a son and grandson who never smelled fear with their own noses, who nonetheless came into the world already sensitive to what their grandfather had learned to dread. We followed the Dutch hunger winter, a famine that ended before its victims' children were born, leaving biological marks in those children's epigenomes that were still measurable 60 years later, and traces, smaller and more subtle, in the generation after that. We followed the research on Holocaust survivors and their children, the altered cortisol systems, the changed methylation at stress-regulating genes, the careful, honest acknowledgement by the scientists doing this work that the evidence is compelling but not yet conclusive, that social transmission, prenatal environment, and germline epigenetic inheritance are three overlapping pathways that have not yet been fully separated in human studies. We understood the mechanisms: small RNA molecules in sperm, methyl groups attached to gene promoters, the signal that travels from the brain of a stressed organism through pathways that are still being mapped to the reproductive cells, and from there to the next generation. we considered the adaptive interpretation, that the biological inheritance of trauma may not be damage alone, that a more sensitive stress system, born of a parent's survival, is a preparation for danger, that when the preparation proves unnecessary, it becomes a burden, but that the mechanism itself is evolutionary wisdom, not evolutionary failure. We learned that epigenetic marks can change, that exercise and therapy and safe experience and enriched environments can write new marks over old ones, that the inherited biology is a starting point, not a sentence. And we sat with the profound and unsettling idea that you carry in your biology the echoes of lives you never lived, that the people who came before you are not entirely in the past, that their experiences, their fears, their famines, their losses may have written something into the molecule that runs your body, and that you carry that writing with you, whether you know it or not. What does this teach us? It teaches us that we are more continuous with the past than we usually understand, that the line between history and biology is blurrier than it looks, that the stories of your family are not just stories. They are context for understanding things about yourself that might otherwise seem to come from nowhere. It teaches us that biology is not destiny. The epigenome responds to experience. The marks can be changed. The inheritance is real, but not immutable. You have a life of your own, and that life is writing its own marks, alongside the ones that arrived with you. The two are always interacting, always updating each other. It teaches us that healing can be biological as well as psychological, that when trauma is addressed, when the nervous system is calmed, when safety becomes real rather than conceptual, something may be changing, not just in the mind, but in the cells. That the work of recovery is not only about the past, it is about creating the conditions right now in which the epigenome can begin to write something different. And it teaches us to hold our ancestors with more understanding. The anxiety you inherited, if you did, was not a failure on their part. It was survival. It was the body doing its job. It was a message sent forward through time, written in the only language available. The language of chemistry, the language of methylation and small RNA, the language of the gene, marked by experience, passed through the sperm or the egg, arriving at last in you. You are the recipient of that message. You did not ask for it. You cannot return it. But you can read it with more compassion than you might have otherwise, and you can begin, if you choose, to write something new alongside it. The ghost in your DNA is not a haunting. It is a conversation, a long, slow, chemical conversation across generations about what the world demanded and what it cost and what survived. You are the latest word in that conversation, and the conversation is not over. There is one more thing worth holding as tonight draws to a close. The science of epigenetic inheritance is still being written. The debates among researchers continue. The evidence is real but incomplete. The mechanisms are identified but not fully mapped. The human studies are suggestive but not conclusive. And the field is young enough that major revisions are still possible. This is the nature of science at its frontier, not settled certainty, but honest investigation of genuinely difficult questions. The scientists doing this work are careful people, navigating the gap between what the animal models suggest and what can be demonstrated in humans. They are aware of the methodological challenges. They are aware that the popular press has sometimes run ahead of the evidence, and they continue anyway, because the questions are important enough to be worth the difficulty. What is already clear, even in the incomplete state of the current evidence, is enough to change how we think. The epigenome responds to experience. Experience is transmitted through biology as well as behavior. The biology of one generation shapes the biology of the next. And that biology is not fixed. It is living, responsive, and capable of change throughout a lifetime. That is enough, more than enough, to sit with and to carry forward, and to let soften into sleep. Now, let all of that become soft. Let the methyl groups and the small RNAs dissolve back into the simpler awareness of your own breath. Let the stress response quiet. Let the inherited patterns rest. They have been carrying something for a long time. Tonight, they can set it down. Now, let all of that become soft. Let the chemistry dissolve back into feeling. Let the methyl groups and the small RNAs and the glucocorticoid receptors fade into the simpler awareness of your own body, breathing, resting, safe. Your ancestors survived what they survived. And here you are, carrying something of theirs and making something of your own. And for tonight, at least, that is enough. Let your body become heavy. Let the quiet settle over everything. Let the long conversation of your biology grow still, for now, and let sleep come in its place. Good night. If this journey left something in you, please consider liking or subscribing. It helps other curious minds find their way here. Good night.